Hello, I am Emily Garza. My partner is Caitlin Torres, and I will be presenting our PowerPoint for cell-mediated immunity. Okay, so the intro. T-cell-mediated immunity combats microbes that are intracellular. These intracellular cells include phagocytosed microbes, microbes that have escaped these vesicles, and microbes within infected cells. CD8 cells, or cytotoxic T lymphocytes, and CD4 T cells, or T helper cells, are responsible for these reactions. So the function of helper T cells. So T helper cells coordinate and regulate the activation of cytotoxic T cells in order to destroy infected cells. It is able to do this by secreting cytokines such as interleukin-2 and interferon gamma. Once it activates the cytotoxic T cells, it also activates the macrophages since they become better at killing infected microbes. Additionally, T helper cells can also regulate inflammation by secreting interferon gamma, TNF alpha, and interleukin-17 cytokines that will then recruit immune cells to the site of inflammation and/or infection. The function of cytotoxic T cells. So, cytotoxic T cells are able to recognize the antigen that is presented by MHC1 on the surface of the infected cell. Once they're recognized, it becomes activated. After it is activated, it goes through a process called clonal expansion that then results in the proliferation of antigen-specific T cell clones. These clones are then able to attack the infected cells. Finally, when cytotoxic T cells release perforin and granzymes, they are able to induce apoptosis, which is the main function of these T cells. So there are 4 different steps in the activation of T lymphocytes. First is antigen recognition. T lymphocytes find a specific antigen that is presented by APCs. Second is activation. Once a signal is sent that activated intracellular pathways, it also activated transcription factors that are needed for the T cell to be activated and as well as proliferation. Third is clonal expansion. Activated T lymphocytes undergo this process and result in antigen-specific T cell clones. And finally, differentiation into effector and memory cells. These activated T lymphocytes differentiate into effector cells with their functions. Once this happens, the effector cells are now able to eliminate pathogens through their received function. Finally, once the infected has subsided, some activated T cells are then left behind and now serve as memory T cells. So there are multiple signals that are needed to activate these T cells. The signals that are needed are the MHC signal, as well as the co-stimulatory signal. So the ligands and the receptors that are used, the T cell receptors are able to recognize and bind to the antigen that is presented by the MHC molecule. The CD4 or CD8 receptors are able to bind to the specific MHC molecule and help better with the interaction between the T cell receptors and the MHC molecule, which will then in turn activate the T cell. The adhesion molecules carry the ability to increase the strength of the binding between the T cells and the APCs. And finally, the co-stimulators. The signal that is sent from this interaction results in the activation of the T cell and proliferation. So T cell mediated immune reactions. So there are 2 types of immune reactions that are from CD8 and CD4. The first is cytokine secretion. Your CD4 T cells activate to secrete cytokines. These cytokines will recruit other leukocytes in ingesting microbes and activate them to destroy these phagocytosed microbes. They will also cause inflammation that will, as a result, kill microbes. And second is apoptosis. The second reaction comes from CD8 cells, which secrete cytokines that will kill infected cells through apoptosis. So there are 4 different subclasses of CD4 cells that naive cells will differentiate into. These are Th1, Th2, Th17, and THF. These subclasses can be separated based on the type of cytokines they will release. So first, Th1. Th1 releases interferon gamma cytokines that target intracellular pathogens within macrophages. Th2 Th2 will release interleukin-4, interleukin-5, and interleukin-13 cytokines that deal with defense against helminths, I believe that's how you pronounce that, and activate eosinophils and mast cells. Th17 will release interleukin-17 and interleukin-22 cytokines that target extracellular fungi and bacteria, as well as recruit and activate neutrophils. And lastly, THF. These release cytokine interleukin-21 that deals with the differentiation of B cells. So there are two mechanisms in which macrophages are activated, classical and alternative. The classical activation, also known as M1, is utilized by interleukin gamma, which is released by Th1. This activation results in activations such as phagocytosis and the killing of fungi and bacteria, as well as a pro-inflammatory response. The second macrophage activation is alternative activation, also known as M2. This is activated through the release of interleukin-13 or interleukin-4 cytokines by Th2. The response by macrophages is anti-inflammatory, supports wound repair, and fibrosis. There is a necessary balance between these two activation types in diseases such as leprosy. And how do CD8 mechanisms of killing infected cells? So CD8 cells, or also known as CTL, cytotoxic T lymphocytes, are the second important cells within T cell mediated immunity. These T cells will recognize the class 1 MHC on the surface of infected cells and kill them. When CTLs are activated after binding to an infected cell, they will release their granule contents containing perforin and granzymes towards the cell. Perforin will allow an entrance for granzymes, and granzymes will enter the cell's cytosol and induce apoptosis, killing the microbe as a result.